So we're going to focus on population ecology today. Okay? Population ecology is going to be anything that's affecting the population size over a period of time. And those could be factors like disease, evolution, just like I said, anything that's affecting the population size over a period of time. One of the things that could do that would be density. Okay? Density is the number of individuals per unit area or volume. And you'll have, think about a population of humans. Okay? If I have a population that's more dense, so essentially the area is more crowded. Because again, for density, I'm talking about the number of individuals per a unit of area or volume. Okay? Remember the slash there is per. So if I'm looking at a place that's more crowded, think about disease spread. If a disease is going to spread more quickly through a dense population, for instance, a crowded city versus a rural population, where people are not coming in contact with one another as easily. Another thing that can affect that is dispersion. Okay? Dispersion is basically just how the organisms are spaced out. They're spread out throughout the population. So remember, your population is going to be the organisms of the same species in one particular area. And so your dispersion is how they are spaced out within that actual population, within that actual area. Okay? And so the first one we have is these penguins here. This is called a uniform dispersion. So in this case, they are spread out in a uniform way. So there's the same amount of space between each organism. These are usually with territorial animals. Okay? This is usually a result of some pretty heavy competition, and they have staked out their territory and nobody's allowed in their little area. Okay? This is usually pretty rare. This is only going to be in an area where environmental conditions don't change. The environmental conditions are basically equal everywhere. So there's no one spot that's better than another spot. So it doesn't matter if everyone is not sharing resources or has their own particular little area here. So uniform distribution, like I said, comes from a lot of competition. So there's lots of competition within the population, and it is relatively rare. You have to have environmental conditions that are pretty much the same, constant everywhere. Okay, the next one is a random distribution. So a random distribution is just that. There's no competition here. They aren't in groups. They're not in clumps. They're just kind of wherever they want. Okay, so there's not a lot of competition. Nobody's staking out territory. They don't necessarily like to form groups. That doesn't help them or benefit in any way there. Okay, so again, you have conditions that are pretty uniform here. Okay, and this is also relatively rare. The third one is clumping. And that's both of these pictures over here, both the bottom and the top there. Okay, so clumping, they form random groups. Okay, you can see the herd here and the flock and the groups of flowers and the school of fish. Okay, so they're forming random groups. And this is going to be due to the environmental conditions. The environmental conditions are not the same. Okay, and so they are grouping together in a way that they can exploit all of the resources in the environment. This is going to favor reproduction by putting them in contact with other organisms of their particular species for reproduction. It can also be a benefit for helping protect the organisms from predators. Okay, so there's a lot of benefit to this clumping distribution. So again, you've got protection from predators, favorable reproduction, both of those being benefits of this. So sometimes when we're looking at this dispersal, we can have a long-distance dispersal. So these organisms have spread out over a very large area, and that can result in what's called adaptive radiation. And adaptive radiation is when the group of organisms start to diversify. Okay, so I get diversification of this particular group of organisms. So, for example, I see it with the finches, Darwin's finches on those islands. Hey, remember you've seen those examples of the finches, and they all have the different kind of beak. Okay, that's a result of adaptive radiation. As these organisms were spreading out over these long distances, they were filling these various ecological roles because they didn't have a lot of competition from the native species. So they were filling these holes basically in the ecosystem. And so you get this adaptive radiation. Again, the species itself starts to diversify to fill these ecological niches because, again, there's little competition from the native species. All right, let's look at it over on this picture here with these honeybees, the African honeybee. So the African honeybee, okay, it first arrived, obviously it's African, so it's not supposed to be here. Okay, it's not supposed to be in the Americas. And it first arrived in the Americas around 1950. Okay, and you can see it first showed up here in Brazil. You see a 1956, 1957 time frame there where they were seeing it in Brazil. Okay, and it took, if you look at the graphic here, how long it took to reach the U.S., it took about 40 years to reach the U.S. Okay, these are the kinds of things that seem like straightforward, obvious things, but you're going to have to be able to occasionally write these things and not be so concerned that that was too easy. They first arrived in Brazil in the 1950s. It took the bees 40 years to move into the U.S. But here's what's important, and what's always important is your why. Why did they move into the U.S. in the first place? Why were they more successful than the regular bees? Okay, first of all, they produced more offspring, so they were better at reproduction. Okay, they're better defenders of their nest. Okay, these are the bees that you hear about, like them stinging people like crazy and killing people. Okay, so they defend their nest a little bit more fiercely. And when they're threatened, they're actually more likely to abandon their nest and go find another place. And so they will abandon their nest and leave instead of defending it to their death. And so that made them more successful, and they were able to travel up through the Americas. And this would be an example of adaptive radiation. Okay, these bees are starting to diversify as they're headed up through the Americas. They're filling these niches that were empty from the native species, so they're reproducing well, okay, and so they are continuing to spread throughout the Americas. So their geographic range now would be from South America all the way into the southern United States. So their geographic range of the organism, again, is going to be the area where you're going to find the species. So some organisms have a very large range. Some organisms have a very small range. Okay? It's just going to be the area where you're able to find the organisms. So if you look at these two particular graphs, one is for North American birds. Okay? And you'll see you've got the largest percentage of species have a very small geographic range size. Same with the plants. Okay? Almost about 45% or so of the plants are going to have the smallest geographic range size, which would make sense that if you look at the scale on the bottom, okay, that the plant range is smaller than the bird range. It is more difficult for the plant to be able to travel, so they're going to be closer together, and they're not going to have the same dispersion and range as a bird that could fly from area to area. So when we're studying population ecology, we may need to have an idea of the population size. Okay? And so there's a couple of ways that we can determine that. The first one is that we can actually count each individual. So we can count the total number of organisms, which if you think about it logically is almost impossible to do. Okay? It would be very difficult, especially with certain species, to really determine whether you got every single organism or not. Another way is to estimate it by counting a portion. So you could divide the area into a series of, let's say you're looking at a 10-acre woods. You could divide the area into 10 different acres and count the individuals on a couple of those acres and then apply it to the entire area. The third is mark and recapture, or mark and capture. And that's what you see happening there at the bottom, okay, is this mark and recapture technique. So with the mark and recapture technique, you're going to first capture a limited number of individuals. So not a bunch, just a random number of individuals. Let's say you captured 20 or 30 of them. So let's say you captured 20 of them. They're captured at random and they're tagged. You can see the tag that's on this crab here. So the organisms are captured at random, they're tagged, and then at a later time you capture a second group of organisms. And what you will do with that second group of organisms is you will first look and see how many of them are already marked. And so you determine the percent of your marked individuals. So you determine if you got, 50, 75, 15%. So you determine the mark of the percentage of the marked individuals. And so you'll then use that to extrapolate out the entire population size. Okay, let's walk through an example. Okay, so let's say I'm practicing the mark and recapture. And my first group, I had 20 individuals that I tagged. And in my second group, okay, I had 10% of them were marked. So this is in my second group. This would then tell me that that 10%, that the original 20 represents 10% of my population. Okay, so the original 20 equals 10% of the population, which then means that my population is going to have 200 individuals. Because if I'm looking at 20 individuals are equal to 10% of the population, well, then that's going to be 200 individuals. There is an equation for this, and we're going to be doing plenty of equations, as I'm sure you saw on your formula chart. It's S over N equals X over little n. Okay, so S is the number marked in the first sample. So S would be, for instance, the 20 that I had. It's the number marked in the first sample. X is the number marked in the second sample. Little n is the number of individuals in the second sample. So in total, how many did you capture? Not how many did you mark, how many are marked. Okay, so number of individuals in the second sample, total. And then N is what you're actually looking for. N is your estimated population size. And you just set it up as a proportion there. You could also rearrange it, but so in our case, we would have the 20 original individuals over N equals our 10 marked in the second sample. Let's say that second sample was 35. Okay, and then when we do the algebra here, we're at a population of 70. And you cross multiply, right? Cross, divide. Okay, and we're at a estimated population size of 70. Right? When we're also studying populations, we'll look at what are called survivorship curves. There are 3 different types of survivorship curves. The first one would be a type 1.